Hi, everyone, and thanks for joining us today for our educational course on time-varying connectivity in resting state fMRI. So this is the first talk of the course, and the aim of this first talk is actually to give you a quite broad and comprehensive overview of the methodological approaches that have been used and that are currently mostly used in literature in order to assess the time-varying behavior of fMRI resting state connectivity. So first, we will actually define the basic concepts of functional connectivity, and then also define the terminology that we're going to use in this educational course in order to avoid confusion. And then we'll dive into the details of the methodological framework. So functional connectivity is defined as the statistical dependence between functional signals that come from different regions of the brain. So in general, we have a subdivision of the cortex in different regions of interest. And what we do is simply computing the average functional signal inside each region. So what we refer to usually as regional time courses that can come, for example, from fMRI. And computing functional connectivity starting from these time courses is very simple. In fact, it means just computing pairwise correlations. And this yields a connectivity matrix, which is an ROI by ROI matrix in which each entry actually includes the correlation for each connection. So this is what we refer to as static functional connectivity. And static functional connectivity has two main properties that I would like to focus on today. The first one is being averaged. It means that it yields one measure over the whole acquisition length. So for example, for the time courses, it gives the correlation raw. Related to the fMRI resting state, it means that the whole acquisition length, which is normally between 5 and 15 minutes, in which a brain volume is acquired every 1 to 3 seconds, would yield just one correlation matrix. So of course, you wouldn't be able to see anything about temporal variations of this functional connectivity, but you would have a picture which reflects a kind of an average behavior of what happens inside the acquisition. The second property is being memoryless, meaning that static functional connectivity is invariant to the reordering of time points. We can take an example, our two time courses that are characterized by the correlation raw. If we actually permute time points, the resulting time courses would be always characterized by the same correlation, because the correlation is a metric that does not take into account the influence between subsequent time points. So time points are considered actually as independent samples. Detaching from these two main properties, being averaged and being memoryless, is what we want to do when trying to go beyond static functional connectivity. So when we want actually to explore the temporal variations and the dynamic features of connectivity, as it has been shown to be important in the last decade. In particular, if we relax the first property, being averaged, we want to find a method that actually shows the temporal variations, so the variations in time of functional connectivity, and we will define that as time-varying functional connectivity. For example, the very well-known sliding window approach, co-activation patterns, and so on are all methods belonging to this first category of methods. Then secondly, if we relax instead the property of being memoryless, we are looking for a method that actually one considers the influence between subsequent time points. And we would call that dynamic functional connectivity. And to this category, for example, we have several methods belonging to that. So, for example, autoregressive models, innovation-driven caps, spectral approaches, and so on. So in this talk, I will give details about the mainly used methods in literature belonging to these two categories, so time-varying FC approaches and dynamic FC approaches. And then last, I will also touch on methods that include or try to include the structural connectivity information in the exploration of functional dynamics, as this is also a very prominent and promising line of research. But first, we start from time-varying functional connectivity approaches, and in particular from sliding window FC analysis, which is currently still the most commonly used in literature. So how does it work? It's very simple. Again, if we consider our functional regional time courses as before, we compute a correlation, but instead of considering the whole acquisition time, we just consider a restricted window, which in general can be around 1 minute. We compute a correlation and then we slide the window for the whole time of the acquisition. What we obtain instead of one measure of correlation is the evolution in time of that value of correlation for each connection. So if we do it, if we look at it at the global level, so at the whole brain level, we obtain a functional connectivity matrix for each window. And this is the information that we refer to as time-varying functional connectivity. Given this framework, we can actually quantify and summarize the temporal features of this functional connectivity in different ways. So the more straightforward way is to compute some metrics of connection-wise variability, which means simply, for example, computing standard deviation or variance of the time courses of each connection to understand which of the connections are more variable. Otherwise, if we look at the whole brain, we can actually perform a time-varying graph analysis in which we compute a specific graph property, for example, modularity or efficiency, and we look at the temporal evolution of that property during the acquisition. Otherwise, if we apply some decomposition technique to the time-varying functional connectivity data, we end up with what we call connectivity states that are patterns of functional connectivity that mostly recur across time in one subject or across subjects if we concatenated the data of more subjects. The property and the interpretation of these connectivity states actually depends strongly on the technique that was used to obtain them, of course. So, for example, K-means clustering was introduced by Allen and colleagues in 2012, and after that broadly used by many people in literature, or PCA was introduced by Leonardo in 2013, but other methods have been used like dictionary learning and so on. And after we obtain connectivity states, we can actually look at their temporal properties in different ways, depending on the features of the states. For example, if our states are exclusive in time, as it happens with K-means clustering, we can actually look at the temporal occurrences of these states. And we can compute temporal metrics, for example, the average duration of each state or the transition probabilities from one state to the other. Otherwise, if the states are jointly expressed, it means that for each time point, we will have a multidimensional description of our states. So states that coexist at the same time point in a macro states that are called metastates and that were introduced by Miller in 2016. So in this case, we can actually look at occurrences of metastates. But then, as I said, the sliding window approach is for sure right now still the mostly used in literature, but it also has pitfalls and limitations that needs to be carefully addressed. One critical point is the choice of the window parameters, in particular the window length. In fact, when we choose the window length, as this can even influence quite largely our results, we should always consider a trade-off between sensitivity and specificity. What it means is that the window should be actually short enough, of course, not to miss the fluctuations in connectivity that we want to investigate. And this can depend on the specific application if we have or not, for example, an idea of the timing of our events of interest. And specificity means that at the same time, the window should be long enough to detect only real fluctuations. Why? Because it was shown that actually spurious fluctuations can be also included due to the window we process. And a rule of thumb in order to avoid that is to keep our window always longer than 1 over the minimum frequency present in the data. And this minimum frequency actually depends on most of the time on the pre-processing steps, and in particular on the high-pass filtering cutoff that we apply to our data, to our fMRI time courses during pre-processing. As to the window shape, the most simple shape that was adopted is the rectangular one, but other shapes have been introduced, for example, in order to ameliorate the signal to noise ratio. For example, the tapered window that allows to smooth a bit the borders, and so actually give less importance to noisy peaks that might be in the borders of the window. Then another critical choice that needs to be addressed is the choice of the parcellation. This actually is not entirely strictly related to, only related to a sliding window approach, but in general to functional connectivity, because many atlases, many parcellations are available based on different criteria. So functional, anatomical, or multidimensional, clusters from ICA, and so on. And even here, studies in literature show that actually the choice of the parcellation can also affect strongly your results. So in an effort to go beyond the atlas and avoid to having to choose a priori a parcellation, we introduced this method a couple of years ago of voxel-wise time-varying functional connectivity, where basically instead of starting from regional time courses, we would start from voxel-wise time courses and then proceed with the sliding window approach as explained. But then here the problem that we have is a problem of dimensionality, as these functional connectivity matrices that would be voxels by voxels are too heavy to be computed or stored. And so the strategy that we chose to adopt is actually to consider the eigenvector centrality of these matrices instead of the matrix itself. So the first eigenvector of each functional connectivity matrix. And we also adopted a mathematical strategy in order to compute these eigenvector centralities directly from the functional data, so from each window, keeping the FC matrix implicit so that we don't face this dimensionality problem. These eigenvector centralities would represent a rank 1 approximation of functional connectivity, as they are the dominant patterns, basically, of functional connectivity, only given by the one dimension, only the first eigenvector. Then within this framework, we introduced the first parcellation of the healthy brain derived by voxel-wise time-varying functional connectivity, because in fact, it's true that we wanted to avoid starting from the choice of an a priori parcellation, but at the same time, we could imagine that the time-varying functional connectivity information was not necessary at the voxel level, because neighboring voxel would carry the same information. So what we did was simply merging together the voxels that showed exactly the same information from the time-varying functional connectivity point of view. And this yielded long-range parcellation of 37 distributed patterns that could be further subdivided into 449 contiguous regions. This parcellation is completely data-driven. It solves the issue of choosing an a priori parcellation from the beginning, and it includes the information of dynamics of function that, of course, we want to consider. Then other methods in literature actually tried to get rid not of the parcellation, but of the window. So going behind the window in a category that we would call frame-wise analysis. So to give you a graphical representation of that, if we consider the time courses, the usual 2 time courses, and we go back to a static FC analysis, what we would have is a very blurred picture of what happens inside, right? Because we would have an average, only an idea of the average behavior inside this long window, I mean, the whole acquisition. If instead we have a sliding window analysis, it's true that we average in a shorter time length, but still what we have, it's a blurred image, a bit less blurred, but still blurred because what is inside the image, it's hidden to us. So if there are changes inside the window, we cannot retrieve them. So frame-wise analysis instead asks the question, so what can we actually extract key time points where there are the relevant events that we want to capture without the need of doing any averaging, basically. And this is the idea behind the technique of co-activation patterns that was introduced by Liu and Duin in 2013, inspired by the point process analysis, and hypothesizing that actually there are brief moments of co-activation and co-deactivation that would be missed as they are brief by a static analysis. In CAPS, the technique that is used in order to select the important frames is based on a thresholding of a seed-bold time course, meaning that we have to choose a region in the brain as a seed, a region of interest, and then we want to select only frames that appear when this region is active. And these selected frames then enter a step of k-means clustering, and the result of this k-means clustering, so the resulting centroids, are what we would call co-activation patterns, the CAPS. And here we show the PCC-related CAPS as posterior cingulate cortex was chosen as seed. And these patterns include regions recurrently co-activating or co-deactivating with the seed for only brief moments, so for a limited amount of time. So we can recognize in these patterns non-resting state networks, but more importantly, these appear also decomposed into multiple sub-patterns that are characterized by a different temporal occurrence, and so that would be missed by a static analysis. Then a second analysis that I would like to briefly mention within always the framework of frame-wise analysis is the LEIDA, the Leading Eigenvector Dynamic Analysis, which was introduced in 2017 by Joana Cabral, who also will give a talk in this educational course, and that basically gets rid of the window by computing the connectivity with an instantaneous coherence connectivity method. How does it work? Basically by selecting the by computing the Hilbert transform of our bold fMRI time courses that yields a bold phase for each region and for each time point. And this allows actually for each connection at each time point to compute the connectivity as the phase coherence between 2 different regions, yielding a connectivity matrix, which is a phase coherence matrix at each time point, so that we can appreciate the temporal evolution. And then similarly to the approach of voxel-wise sliding window correlation that I described before, even here, in order to reduce dimensionality, as we would have 1 matrix per time point, what I do is just approximating the information of the functional connectivity matrix with its first eigenvector, so the leading eigenvector, that yields the rank 1 approximation that you see here, so a quite good approximation. Then going to dynamic functional connectivity approaches, so in order to remind you approaches that are not memoryless, so that want to look or model the influence between subsequent time points. The first one that I would like to describe is the revisitation of co-activation patterns with a dynamic flavor, so innovation-driven co-activation patterns. These ICAPs were introduced by Calhoun and Van de Ville in 2015 within our lab. And the flavor, the dynamic flavor, is given by the fact that it's not functional signals that enter the clustering, but it's innovation signals, where innovation signals are defined as the first derivatives of denoised and deconvolved signals. So computing the first derivative means that you're looking at the change between subsequent time points. So it's the transitions in the bold signal rather than the peaks that we are interested on. And so voxels that cluster together here are voxels that change their level of activity, so increase or decrease their bold signal together, so independently of the initial level of activity. And similarly to CAPS, the clustering steps yield here a brain repertoire of functional networks, where we can retrieve again the most common and known resting state networks. For example, the default mode network is still the ICAP with the longest duration. But also, more importantly, we can also have access to subsystems of the default mode network, because you can see that also patterns are retrieved where only sub-portions of the DMN appear together with other regions of the brain with which they synchronize only for a brief amount of time. So this information would be missed by static analysis. Then regarding temporal modeling, one possible method that performs the temporal modeling is the autoregressive models. Autoregressive models actually model the fMRI volume at time point t as a linear combination of the fMRI volumes at p previous time points, p being the order of the model. Here, A, the matrix AL, is the matrix of coefficients that we need to estimate and contains the dependencies between a time point t and a previous time point t minus L. And for that, it can be seen as a dynamic version of the correlation. And here, autoregressive models are interesting in this context because it was shown by Raphael Liegeois, who will also have a course, a talk in this course, that first-order AR models can explain FC fluctuation better than static functional connectivity and then hidden Markov models. And then more recently, he showed actually that the dynamic functional connectivity markers obtained by AR model together with static how they can be linked to behavioral measures. So basically what emerged from this analysis is that on average, dynamic measures of functional connectivity explains more behavioral variance than static measure. But more interestingly, it seems that static and dynamic metrics are actually complementary because they explain differently different behavioral metrics. For example, task-based phenotypes that are characterized by network interactions at faster timescales are better captured by dynamic metrics and are missed by static functional connectivity metrics, while other type of behavioral phenotypes, like self-reported measures that are trait-like properties that are not characterized by these fluctuations at high frequency, are equally well captured by static and dynamic functional connectivity. Then autoregressive models were employed also in order to retrieve the first dynamic extension of component analysis, dynamic mode decomposition. And in this case, it's the matrix A that is decomposed, eigendecomposed, yielding modes that contain a spatial information given by the eigenvectors of A and a temporal information given by the eigenvalues of A. So these spatiotemporal modes that are obtained are characterized by a specific damping time delta and a specific period of oscillation T. For example, we can look at the first two modes obtained in the paper by Casorso et al. In 2019, and these two have different temporal properties. The first one actually contrasts two very well-known resting state networks, so default mode network and task positive network. And the second one, interestingly, put in opposition is only a subportion of the DMN, showed in red, with the visual network. So again here, as it happened for the ICAPS, it shows that taking into consideration, taking into account the dynamic feature of functional connectivity allows to actually have access to subsystems, in this case of the DMN, but in general of resting state networks. Then last, I would like to mention, as I said, a line of research that I think being very important and promising for the future, and it aims at integrating the structure in the analysis of functional dynamics. Actually, the question here is how different functional patterns, functional connectivity patterns that actually change over time can be generated, can emerge from the same static structural connectome, structural connectivity. And I will touch on two different frameworks that address this question. One is graph signal processing, and the other is whole brain computational models. So graph signal processing is an emerging field that has been only recently applied to the brain for the integration of structural and functional information. And in this case, we have a graph which in this context is given by structural connectivity. So the physical, reflecting the physical links, connections of white matter connecting different regions. And we have a graph signal, which is a signal that is defined at the very same nodes of the structural connectomes of the graph. In our case, the signal is given by the functional activity and changes over time. So the question is, how is the function using the underlying structure? And in order to address this question, the first thing that we could do is actually decomposing the structural connectome into its structural harmonics that were introduced by Atasoy in 2016, and that correspond to the eigenvectors of the Laplacian of the structural connectome matrix, and conceptually could be viewed as a structural backbone of the brain. Each structural harmonic is characterized by a specific spatial frequency which is increasing. So as you can see, the first one is 0, meaning that we have no spatial variation in the pattern, and then increasing frequency of spatial variations. What we did in a work of last year in order to put in relation function and structure in this framework was to try to explain at each time point the functional signal as a linear combination of these structural harmonics, so of the structural patterns. And then we wanted to filter the low frequency patterns and the high frequency one. So decomposing the functional signal at each time point into two portions. One that only contains low frequency, that by definition is more coupled to the underlying structure because of the characteristic of the features of these patterns, the low frequency patterns, and the other portion which is more decoupled. And we could also define a structural decoupling index that quantifies the coupling between structure and function at each region in the brain by the ratio between decoupling and coupling. So if we look at this structural decoupling index that is shown here, we can see actually that the decoupling structure function changes across the brain depending on the specific functional network that the region is on. For example, for lower level primary sensory regions, it appears that they are most often more coupled with the structure. So here represented in blue, while higher level cognition regions appear to be often more decoupled with the structure, so towards red here. And this structure function coupling actually has also behavioral relevance, which is shown by this neurosynth meta-analysis that yields a macro scale gradient going from the lower level behavioral domain to higher level ones when we actually go from more coupled nodes, so the ones in blue, towards more decoupled one from the structure, the one in red. But now the question is, does the temporal variability of this structure function coupling also relate to behavior? So in a preliminary work always developed in our lab, this question tried to be addressed by computing the standard deviation and instantaneous change of the coupling structure function and used to predict 68 behavioral domains. So what was found out is that actually for several domains, dynamic measures in the form of the standard deviation and instantaneous change could actually significantly predict the significant prediction accuracies which were much higher than it was possible to reach with static measure. So still a preliminary result, but very promising for future analysis. Then last, the framework of whole brain computational models also tries to integrate the structural connectome in the analysis of functional connectivity dynamics. In particular, whole brain computational models try to simulate the functional signals in the brain, starting from anatomical constraints given by the structural connectome and models of the neural dynamics. And even if many approaches exist in literature, many different generation approaches for these models, what became clear in the recent years is that simulated data should not only satisfy some spatial criterion, meaning that of course the FC spatial patterns should be reproduced, but also they should satisfy spectral and temporal criteria. So it is very important, it is essential to incorporate the information about dynamic functional connectivity in the model, in the construction of the model, in order to simulate functional signals that are realistic, and more importantly, in order to investigate the real mechanisms of generation of functional dynamics, starting from the structure, the underlying structure. So with this, I want to conclude, and I wanted to give as take-home message, I wanted you to leave with the fact that the brain, even during resting state, is always active and dynamic. Functional connectivity is dynamic, it changes over time, and we need to adopt specific methods that actually look at this feature in order to analyze it. There is a plethora of methods, and the choice of methods always depend on a trade-off depending on the needs of the specific study. Choice between data-driven or model-based methods, or dynamic or time-varying, and windowless methods are still underused in literature, especially in the application to clinical context, but do hold great potential for the future, and so does the integration of structural connectivity. So with that, I thank you for the attention. And I wanted also to take the chance to thank my great colleagues in MIPLAB in Geneva. And this is a picture of us at HBM 2019 in Rome with very happy faces. And I wanted to leave you with this and with the wish of meeting you all again in person very soon in another HBM meeting. Thank you.
So hello everyone, my name is Raphael Liegeois, and I'm happy to welcome you to this second talk of the Dynamic Functional Connectivity course on null models of fMRI time series. It's the first year we have a presentation on this very specific topic because null models have gained a lot of interest recently, and especially in the context of DFC studies, and we thought it would be interesting to browse through the theory underlying null models, as well as on how we can interpret the results of null models, because you will see that it's not always that easy to have a finite or defined interpretation of the final results. So without further ado, let's get back to the theory of null models and just conceptually explain how it works. What are we doing when we do null model hypothesis testing? Well, essentially it consists in trying to understand the nature of the statistical information that is contained in some data that we are using. So typically here we have a time course and we are wondering what is the statistical nature of this data. So starting from this, we denote the original data. The original data, of course, null model testing consists in identifying a model, big M, from this data set. Using this model, we can generate what we call surrogate data. And then we will define, and using a metric called small m, that we can compute in the original and the surrogate data. And then by statistically comparing the values of m in the original and the surrogate data, we can have some conclusions about validity of model m. So for example, if MO, so M, the metric M computed in the original dataset, is different from MS, which is M computed in the surrogate data, then it means that M, the model M, is insufficient to capture all the statistical information that is contained in the original data. On the contrary, if MO is equal to MS, then it just means that there is no evidence that big M is insufficient to represent the original data. So in practice, it means that we will go for this model. So this is conceptually quite easy, but you will see that there are some intricacies that needs to be paid attention to when interpreting the results. Let's consider a simple example, which is the univariate Gaussian model. So what is the model that we test when we use the univariate Gaussian model? Well, essentially, we assume that each time point of the original data has been generated by a Gaussian with a mean mu and a standard deviation sigma. And this is the case for each time point. Then using this model, as I said on the previous slide, we can generate surrogate data. And so I'm not mentioning anything about operation on the data. You will see in the next slide why this line is used for. So let's here take a step back and think of which statistical properties are being preserved in original and surrogate data. In this case, it's quite simple. And it's essentially the mean and the standard deviation of all the time series here. By construction, these values, the mean and the standard deviation, are the same in the original and in the surrogate data. An example of test that we could do, for example, in this case, to compare original and surrogate data is compare, for example, the counts of threshold crossing. So M would be the number of threshold crossing. We could also compare kurtosis or any higher order statistical measure that is not defined by the mean or the standard deviation, or any other metric, again, that is not defined by mu or sigma, because these are by construction the same in original and surrogate data. So it doesn't make sense to compare these two. We expect these two to be the same. Now, how can we conclude about the results that we have from this test? In the case where MO is different from MS, this means that data cannot be fully explained by a Gaussian, by a univariate Gaussian. In other words, this means that mu and sigma are not sufficient to fully characterize the data, and there is some statistical information beyond these two metrics. If MO cannot be shown to be different from MS, then it means that there is no evidence that data cannot be explained by a Gaussian. That's what it means. So this was a very simple example. Let's now consider a slightly more elaborate example. So what happens when we generate surrogate data by shuffling all the time points? For those who are a bit familiar with the null model testing literature, this is a very common procedure. But what exactly are we doing in this case? So this will be a first question that I invite you to think of for a few seconds. So are we testing the same model as in example 1, but using another procedure? Are we testing the fact that data, the original data, is IID, meaning independent and identically distributed, with a distribution that is matched to the original one? And C, the fact that data follows a first-order autoregressive model. What do you think? Well, let me give you a small clue. Consider here this curve, which represents the distribution of the original time points. It should be quite obvious that by shuffling all the time points, this distribution is preserved. And so based on this, the answer is B. We are testing that data is IID with a distribution matched to the original one. So in mathematical terms, we are testing that each time point xt follows a distribution D, which is the one of the original data. What are the data being preserved, data properties being preserved in this case? Well, essentially, all the statistics that can be computed from D, the distribution of the original data, are preserved. So in particular, the mean and the standard deviation are preserved, but also the kurtosis and any higher order, for example, statistical measure. What are meaningful tests that we could use to compare original and surrogate in this case? Well, we could compare again the counts of threshold crossings, compare autocorrelation in the original and surrogate, because autocorrelation will be destroyed in surrogates, or any other metric again, but nothing involving the distribution, or nothing that is defined by the distribution of the original data, because this is by construction preserved in the original and surrogate data. Again, the conclusion in this case is if MO is different from MS, whatever the metric you are using, whatever metric you are using, this means that data contains some form of autocorrelation, meaning that successive time points are linked and there is some information flowing between successive time points. If MO cannot be shown to be different from MS, then it means that there is no evidence that temporal ordering is important. Okay, so this was for the conceptual sketch. It is, I think, quite simple using these, considering these simple examples, but you will see that the interpretation of the results, as I said, can sometimes be tricky. So, for example, a few points that I wanted to make here. The first is the fact that null hypothesis testing is different than testing for relevance of a result. What null hypothesis testing is doing, it's involving a statistical test. So you have a p-value, you have everything, but you are testing that your measure or your metric is exploiting a certain type of information. That's all you are testing. But rejecting or not rejecting a null model should not be taken as a rule to justify that the result is meaningful or not. We will see that some metrics that are that correspond to a model that is easily rejected for fMRI data are still meaningful from a behavioral point of view, from a neurological point of view. Then a second point that I wanted to make is about stationarity, because this notion has gained a lot of attention also recently due to the rise of DFC studies. And actually, stationarity is a statistical property that is always contained in the null model that is being tested. In the two previous cases, stationarity was also assumed. And so non-stationarity is also a potential cause for rejection, but a very non-specific one. And I actually don't know any null model testing specifically for stationarity and nothing else. So I would say don't pay too much attention to stationarity, but likewise, you should not interpret the results of a null model testing in terms of stationarity or non-stationarity only. And finally, and most importantly, I would say, rejecting a null model doesn't mean that the model is useless. To illustrate this, consider the following example that shows the distribution of US women hate in the US population. Horizontal axis is the hate, vertical axis is the proportion of people in each bin. And let's now test whether this distribution can be described by a univariate Gaussian. So this is example one that I just presented. So what we do is we identify this univariate Gaussian from the data. We have this curve with a mean of 64 and a standard deviation of 2.7 inches. And let's now use a metric, M, that counts the number of individuals that are essentially very tall, larger than 75 inches or 190 centimeters. If you do that test, you will find that MO is different than MS because in reality, there are more tall people, tall women than predicted by this univariate Gaussian. But still, the point here is to make that I think we would all agree that the univariate Gaussian is essentially very useful and very close to the actual distribution. And so this means that having one metric that allows to reject a null model doesn't mean that the null model is useless. So we should think here more in terms of distance between a model and the actual data distribution or data structure. Okay, so this was for the first part that was not related to fMRI. Let's now move to the case of null models that were designed to test for the statistical properties of fMRI time series. What is the difference between this case, the case of fMRI time series, and the case that I just presented before? Essentially, it's the same. Conceptually, it's the same. It's just going to be a bit more difficult because fMRI data is multivariate. And so instead of dealing with univariate models and statistics, we'll have to deal with multivariate models and statistics, which makes the math a bit more complex, but conceptually, the testing framework remains exactly the same. So let's go through a few selected null models of fMRI time series. The first one is the multivariate Gaussian distribution. So this model assumes that each time point, again separately, has been generated by a multivariate Gaussian with mean mu. Here we assume that it's 0 because for fMRI time series, we don't really pay attention to this value, and a covariance structure or correlation structure sigma. So this is the case for all t. Using this, again, we can generate some multivariate surrogate data, and the data being preserved here is, as in the univariate case, the mean and the covariance structure. An example of test that we could perform here to test the difference between original and surrogate data is, for example, to compute M defined as the average one lag autocorrelation across all variables, so across all ROIs. What will be the expected outcome of this test? Will it be that MO is equal to MS because autocorrelation is preserved by M? Is it that MO is different from MS because autocorrelation is not preserved by M and fMRI time series are autocorrelated? Or is it that MO is expected to be equal to MS because fMRI time series are not autocorrelated? So here the trick is to notice that when generating surrogates have absolutely no autocorrelation because the successive time points are generated independently from each other. And so the correct answer is B, right? Because we know that the original data, fMRI time series, have autocorrelation, whereas the surrogates does not. Conclusion here is that data cannot be fully explained by a multivariate Gaussian, meaning that if we just stick to the correlation or the covariance as a statistical measure of the information in fMRI time series, well, we are missing some statistical information. Again, and this refers and echoes the point that I made in the warnings at the end of the previous section. This does not mean that sigma or the correlation matrix is useless, and there is infinite literature on that. Okay, let's now go to a slightly more complex null model, which is the one that assumes that fMRI time series are generated by a first-order autoregressive model. The model writes as follows, and so it assumes that successive time points are linked as this, where X is a vector, and so A is a square matrix of size 400, for example, if you are using a parcellation of size 400. From this, again, you can generate multivariate surrogate data. And in this case, what is the data property being preserved? And it is actually not that obvious to really see which property is being preserved. I'm not going to enter the details of how this can be shown, but this is something that we detail in our 2017 paper. But using Euler-Walker equation, it can be shown that this model essentially preserves this statistical information, the mean, the covariance, and R1, which is the one lag covariance structure. So one lag autocorrelation structure. Essentially, it's encoding the link between between all pairs of successive time points. Example of test here could be, for example, examining the amplitude of sliding window correlation fluctuations, or we could also identify, let's say, 5 states in the original and surrogate data and compare the transition probabilities between the states in original and surrogate, and have this as a metric to compare, to test for the null model. So the first point is something, again, that we did in this paper, and essentially what we found is that it's not that easy to reject this null model and to find a statistical difference between the amplitude of the sliding window correlations. So here, what you see in the bottom is some pairs of sliding window correlations. The red curve is the average one, and you can see that in original data, you have some fluctuations, as has been shown by many authors. But in surrogate data generated by a first-order autoregressive model, you also see these fluctuations. Whereas if you use a multivariate Gaussian, typically you would have something that is more or less flat. Okay, so the conclusion here is just that the AR1 model is harder to reject. You will always find some metric that allows you to reject the null, but we are getting closer to the to capture the statistical information in fMRI time series. Let's now consider another quite common null model, which is phase randomization. So again, I'm not going to enter the details of what phase randomization is doing. Essentially, it consists in taking the Fourier transform, the multivariate Fourier transform of the data, keeping the amplitude and shuffling completely randomly the phases, but changing the phase in a random way, similarly across all ROIs. And this is to preserve the covariance structure. For details about that, you can see our 2017 paper or this original Pritchard and Taylor paper that defined phase randomization like that. So this is sometimes referred to as consistent phase randomization in the recent DFC literature. So in this case, again, it's not straightforward to show this, but using the Wiener-Khinchin theorem, it can be shown that the statistical information being preserved by surrogates is the following. It preserves the mean, the covariance, and the complete autocorrelation structure. So R1, R2, etc., until RT, where T is the number of time points of the data. And based on this, it can also be shown that actually what is being tested for is that data time series, fMRI time series, are generated by a linear and Gaussian model of high order, where that is limited by t, where t is the number of time points. It can be 100 or 1000 in the case of HTTP data, for example. An example of test that we could do here, well, essentially it's the same, and that's also something that we did in our 2017 paper. And other people have also shown that it is harder to reject this null. As compared to the multivariate Gaussian, for example, the difference is striking. It is really much harder to reject this null model. So this is a summary slide where, for example, one point to notice here is that these three nulls are nested in terms of the data properties that they preserve. The multivariate Gaussian is easily rejected essentially because it doesn't preserve the autocorrelation structure. But then higher, once some autocorrelation information is preserved, it can be R1 or more orders, then the null is harder to reject. Let's now move to the final part of the presentation that discusses more how to interpret the results of null model testings for fMRI time series. Essentially, what a lot of people, including myself, are wanting to answer is, do my results need to be different on original versus surrogate data in order to be relevant from a neurological point of view, for example, or interesting or meaningful, etc.? And my answer to that, and I know it might be somehow might be discussed, but my answer to this is no, and it might contradict some statements that we've seen in recent literature. But let me explain why I'm answering no to this question. Let's represent the statistical information contained in fMRI data by this big green circle. This is the statistical information that is captured by a multivariate Gaussian model, essentially by the covariance of fMRI time series. This is the covariance plus R1, so the first order lagged information, and this is the covariance plus the full autocorrelation structure that is preserved by the phase randomized null. Let's now consider two cases. The first case is that results that I obtain using a given metric are the same on original and AR1 surrogate data. What does this mean? Well, this means that my method, the metric that I'm using, the method that I'm using exploits information that is within this, for example, this red dotted ellipse. And that's all what it means. It doesn't mean that it's interesting or not interesting or whatever. It's just some information about the statistical information exploited by the method. Consider now another case where results are different on original and phase randomized surrogate data. So which curve could correspond to this situation? Is it curve 1, 2, or 3? So if you are following me until here, I hope you got that in case 2, it means that some statistical information of the outer green circle has been used. Okay, because that's the one that is not included in the full in the statistical information preserved by phase randomization. So correct answer is curve 1. And so again, the conclusions of this is just to say that in case 2, the method exploits information, some information, maybe not only that information, but some information that goes beyond the one preserved by the phase randomizer. And so in these two cases, the conclusion is just to say that we have some information about the statistical information used by our method. This being said, I agree that we could discuss different aspects relating to this, but essentially what I see quite often in papers or also reviews is the fact that people ask to show that the results are different in original and phase randomized surrogates, for example. And I think this is not required for a result to be interesting or meaningful. So the final remarks are the following. as I said, I see that sometimes we require results to be different in original and surrogate data in order for them not to be trivial. This is true to some extent because I agree that in that case, it means that data essentially captures linear and Gaussian information. But let me just remind you that based on what we have seen before, we can now say that the space that is covered and that is described by a linear and Gaussian model of high order has t+1 times n square parameters, where n is the number of ROIs or number of voxels, and t is the number of time points. So if n is typically equal to a few hundreds in an atlas-based analysis, and t is equal to 1,000 and 200 for HCP data, this means that the space in which we are navigating is huge. And so the model is not that trivial, in my opinion. Then another question, which is here an open question, is are we closer to the situation on the left or closer to the situation on the right? Meaning that is the linear and Gaussian structure model capturing a lot of information of fMRI time series or not that much? And my impression is that we are closer to the left configuration because, as I said, ourselves and other authors have shown that it's not that easy to find a metric that is strongly rejected, that strongly rejects the phase randomized null, for example. You will always find such metrics, but again, it's not that easy. And a final, also open question is, okay, maybe there is some additional information beyond the linear and Gaussian structure, but is that information relevant from a neurological point of view? Because in the end, that's what we are interested in. It's knowing more about behavior, about neurological processes, and maybe this outer green curve area is just capturing, I don't know, some strange statistical noise coming from the scanner or things like that. This is an open question, we don't know. And with this, I would like to summarize the main take-home messages of this presentation. The first one being that null model testing allows to know more about the statistical information of the data, but this should not be used to show that some results are interesting or not interesting from a neurological point of view. So in other words, I think it's not useful, it's not meaningful to ask someone to show that the results are different in original and surrogate for the results to be interesting. Then a second point that relates to the first one, rejecting a null does not mean that the corresponding model is useless. We've seen that with the univariate Gaussian case for the distribution of women hate. It's the same for fMRI time series. It's not because we reject, for example, the phase randomized null that a linear Gaussian model is not useful to describe fMRI time series. And finally, there seems to be some converging findings suggesting that the linear and Gaussian structure captured by AR1 and phase randomized null is not that easy to reject. Of course, there is some information beyond these nulls, but it seems that it's not that easy to reject, and therefore linear and Gaussian nulls seem to be a promising way to explore this statistical information going beyond the classical correlation or covariance matrix that corresponds to the multivariate Gaussian null. With this, I would like to thank you for your attention, and I've put here the references that I mentioned in the presentation. Thank you very much.
Hello, it's a pleasure to present today. I'm going to be focused on time-varying connectivity in the context of data-driven approaches and also some clinical applications. I'll cover 5 main areas, starting with an introduction to several approaches, then validation, then I'll talk about clinical applications, some more advanced approaches, and then finally discuss some software tools. So what I'm going to be focusing on today are primarily approaches that use independent component analysis to extract brain networks and their time courses, and then look at the correlations between those time courses. And so we can do this in obviously a static way here on the left, or in a dynamic way. And this is a simple approach just using sliding windows. So we essentially compute dependencies between component time courses as a function of time or window. Shown on the left, you can see a couple individual subjects and how these patterns vary over time. In the top is illustrating what we're trying to capture, which is you can, instead of computing a single correlation here, for static connectivity, we want to capture the dynamics. So here we see strongly coupled time courses at the beginning, weakly coupled at the end, as a simple example. Once we compute those windows, then we have a lot of data. We have to further reduce that down and summarize it. And we can do that by doing clustering, for example. So here we've got k-means clustering to get obtain 5 different states. Then we have now, for each time point, we have a state associated with that time point. And so for any given individual, we can look and see which state they're in at each point in time. So state 3, state 2, state 4 for this particular individual. Here's an example from some data looking at schizophrenia versus healthy controls. You can see 5 different states here. On the top are the healthy individuals. On the second row, schizophrenia patients. Below here are the cell-wise differences. One thing you notice is that the differences cluster differently depending on which state you're in. So for example, state 2 and state 3 have strong relationships, interdependencies between subcortical regions and sensory motor areas. But that's just the start of what we can do. We can also look at things like the dwell time. How much time does an individual spend in that state once they've entered it on average? So here we see states 1 and 2. The healthy individuals occupy significantly more time in these more modular states, whereas the patients spend much more time in these weakly connected states, state 4 and state 5. But that, of course, is just looking at and assuming a single state per time point. We can also look at this in a more continuous way. One way that we've done this is to look at metastate metrics. And so here what I'm plotting is state 1 versus state 2, and there's an amplitude associated with how strongly they're participating in that state at any point in time. Here you can see the profiles of trajectories of 2 different individuals. We can then quantify this over time using a vector called a metastate. And so that vector tells you for the first state what the amplitude is, and for the second state what the amplitude is. And we usually quantize these into, for example, 8 different levels. Once we compute the metastates as a function of time, we can summarize those in various ways that provide some intuition about what's going on. So for example, the number of distinct metastates that are occurring, or the total distance traveled in that space. So this is more of a global measure. This is looking at a summary across all of the different states. One of the things I like about this is that you can compute states themselves using various approaches. We can cluster, as I mentioned, using k-means. We can use principal component analysis or temporal or spatial ICA. And if we compare the number of distinct metastates, or all 4 of these measures actually, between patients and controls, we get similar overall indicators telling us that we've got reduced dynamic fluidity, reduced dynamic range in the patients. So regardless of which type of clustering we use, which give very different looking states because they're optimizing for different things, we get a similar global measure here, at least in this case. We can also look, instead of just at connectivity, we can look at activity over time. And so one simple way to do that is to compute the windowed amplitude of low frequency fluctuation. So you can compute a windowed spectra, compute the low frequency power as a function of time. And now you have that for that time course for each individual subject, and you can cluster those into ALF states. And then this gives you a way to sort of compare how the activity changes over time with how the connectivity changes over time, as one example. And there are a lot of interesting things there to look at. For example, we find sometimes strong coupling between the two, and then we find decreased coupling in other cases. But of course, that is not the end of the story. Functional organization evolves over time, as we know, but I've been talking about temporally dynamic approaches here, where we assume a fixed spatial region and changes in coupling over time between them. But you can also imagine a spatial dynamic approach where the nodes actually themselves are moving around, but the coupling between them is staying the same, or maybe they're shrinking, or maybe they're growing. And then a full spatial temporal dynamic approach where both of space and time is changing. Just to give you an example of what that looks like, one way to get at that is to go to a higher model order to identify smaller basis sets of dynamics, spatial dynamics within a certain domain. So here we have in the middle thalamus, and it's made up of 5 different spatial domains, spatial basis functions, and the weighting of those changes over time. And so you can see how those spatial patterns evolve. Of course, this is still assuming that at the fine-grained level, we've got the ability to capture fixed spatial patterns. You can also remove that assumption and just look fully at spatially fluid dynamics. For example, computing full voxel-wise maps that change over time. If we do that, then we can look and we see really interesting things. We see a lot of variability in certain regions and not in others. We see overlap between states where different regions will merge with other domains and then disconnect spatially over time. And we can then compare these also with various metrics. I'm not going to go into detail on the bottom here, but it's like a spatial histogram, and you can essentially compute a metric from that and summarize it. And the details are in this paper below. Another thing to keep in mind is that most of the work to date has not really systematically characterized whether it's temporal, spatial, or spatial temporal. Everybody will say spatial temporal. But if you look in detail, that's not the case. And so this is our attempt to try to summarize that. I would also preface this by saying that almost any approach you use can be modified to look at the data either temporally, spatially, or spatial temporally. So keeping that in mind, I do still think it's important to try to be clear about what type of approach you're using. In terms of validation, I'll cover that fairly quickly. One approach that we've used in our group is to look at a large data set, 7,000 subjects, to get at replicability. We divided them into 28 different groups, run our full pipeline on 250 subjects in each group, and then compared how similar the states looked across those different analyses. And we find that they were highly consistent, correlations of above 0.7. Interestingly, also that the amount of time spent in the states was consistent as well, showed some similarities. So for example, state 3 was the most highly occupied across all the different analyses. Of course, we're always interested in being careful about motion. We've done some standard motion correction and control on these data, but we also were interested in looking at whether these explicitly dynamic metrics like metastate measures would be, how variable they would be across individuals and to motion. And we found that they were actually pretty robust. Here you can see the distribution of those 4 different metastate measures across the different sets of data. Because those are dynamic, if you disrupt the dynamics using some type of phase randomization, for example, you completely disrupt the distributions as well, which is what this figure is showing. We can also try to get at what these mean by looking at multimodal data. So here's a concurrent EEG fMRI study, and then we can quantify, for example, drowsiness by looking at the alpha power and the delta theta power, for example. And if we order the fMRI states by these drowsiness measures measured from the EEG, we can see that on the least drowsy side, we have high anti-correlation between domains, and on the right side, we have almost a hypersynchronization. So drowsiness is clearly playing a role in some of these dynamics. We can also use this to quantify and predict sleep state. This is some data from Enzo Tagliazucci's group, where they actually quantified a sleep hypnogram over time in an extended resting fMRI study. So individuals fell into sleep, different sleep states, and we were able to get a fairly high accuracy in predicting those sleep states using the dynamic connectivity measures. Here we're getting about 75% accuracy or so using a windowed sliding window approach. Interesting though, if you look at only, say, the most awake state, we also find highly predictable states embedded just within that state. So it's clearly not just sleep that's driving these measures or drowsiness that's driving these measures. That's a factor, but not the only factor. We also know that task is driving dynamics quite a lot. So this is from Javier Gonzalez Castillo's group at the NIH, and so they've looked at blocked task measures. If you use a dynamic connectivity approach to extract components and look at states, we get almost a perfect categorization of the task itself. Again, we can also do sub-clustering within task as well to get at variability there. Here, what I'm showing you is looking at prediction. So we found quite consistently that we get higher predictive accuracies, cross-validated accuracies in a variety of different groups when we're looking at dynamics as opposed to just static measures of connectivity. And so, for example, here, if we take this functional network connectivity matrix and we classify schizophrenia from bipolar from healthy controls, we get about a 60% accuracy. If we use the dynamic states, we bump that up to about 85%, and it's 89% combined. And this makes some sense because you're doing a better job at characterizing the variability in the data within the dynamics, then moving that into a classification framework. Another question that comes up quite often is how many states should we estimate? There are a few ways to think about that. If we focus just on the bottom, one approach that has been used quite a lot is, for example, the elbow criteria. You can try to estimate the number of states there. We did an analysis of 24 different indices, including the elbow criteria, using simulated data. So here, for example, is an 8-state simulation that's designed to look a little bit like a 2-state solution. And what we found were 2 metrics in particular that performed the best: the Davies-Bolden metric and the Ray-Turi metric. All of these are listed in this paper below, which should be online at this point. So I would encourage you to look at that if you want more detail. You can also look at classification as a criteria for determining the number of states. How accurate are your cross-validated classifications given given the accuracy? So here you can see going from 1, this is static, so that's really just 1 state, up to 2 gives you the biggest bump, right? So it bumps up about 5% on average. But there is still some variation in here. We see in this case 4 is giving us the highest predictive accuracy. And that works pretty well. But we can also ask, well, maybe there's a subset of the states that are useful for classification and others are not. So we can do additional feature selection in a greedy approach, and we can see even higher accuracies if we go up beyond 4 or 5. And so what I'm showing you on the bottom here are sort of coming from this plot, something similar where we're using all the states. And what you see here is when you do a feature selection step prior to the cross-validation, where you select a subset of states that are most predictive and remove the others. Here's an example looking at mild traumatic brain injury, where we compared the accuracy of prediction, whether these individuals had mild TBI or not, using fractional anisotropy, static resting fMRI, or dynamic rest fMRI. And you can see the performance is quite a bit higher with dynamics. When we look at the features that were relevant, we see it was these sort of long-range connections between cerebellum and sensory motor areas that were the most predictive. This is some data from Lucina Uden's group and Jason Nomi, looking at executive function in the human connectome data, and they found, for example, consistent links between certain states and certain executive function tests. So, for example, here in this red case, you see for the first half and second half of the data, as well as the entire data set, it was highly consistent and significant. This is just a slide to mention that if you simulate dynamic data, just to be cautious to do it of the limitations of the simulation. So for example, I think it's less useful to try to design a simulation that can sort of exclude the possibility of dynamics and sort of say, make the claim that dynamics doesn't doesn't occur, in the data statistically, because given any model, you're going to have cases that fall outside of that model. So within the context of that simulation, it might be true, but the broader case, I think, needs to be kept in mind. One measure that we've used to try to help us assess the degree to which dynamics are occurring is something called the multivariate phi here. And we find that in a simulated case where we have epicless data, Gaussian random data, real data, and highly non-stationary data, we see in all cases that the multivariate phi is identifying those dynamic scenarios in a way that makes sense. Whereas if you use a measure that's focused on outliers like kurtosis, it doesn't do quite as well there. Okay, now let me cover a few clinical applications. First here is a data set using different subtypes of mental disorders, looking at schizophrenia, schizoaffective disorder, bipolar disorder, and healthy individuals. And you can see 5 different states here. And what I'm showing you in the boxes are the degree to which the groups are similar to one another. So what it's showing us, for example, is consistent with what we might expect. Schizophrenia and schizoaffective disorder are the most similar, followed by bipolar, then healthy controls. Here's just one example of state 4 for these 4 different groups, showing the different patterns cell-wise. In ADHD, this is an example to kind of highlight the fact that these data are complex and you have to be careful in how you interpret it. So here we have a global measure. This is a metastate measure. And if we look at differences, we see increased dynamism in number of states, in the distance, the L1 span distance. But if we look at dynamics within, say, just the default mode network, which has multiple subnodes associated with it, we see reduced dynamism in there. And it's really because you're looking at the data at a different scale. It's certainly quite feasible and plausible that you would get a different type of dynamic behavior. So just keep that in mind as you do this sort of work. This is an example from a normative data set from Tanya White in the Netherlands, the Generation R data set. So we have 800 children aged 6 to 10. I'm showing you 4 different states estimated. They're rendered here on a surface brain. And what we find, a couple sort of observations, is that the older subjects tend to occupy the globally modularized state. Younger kids are more in this globally disconnected state. Interestingly, if we also look at the social responsiveness scale as a measure of autistic tendency, we see that those kids with lower scores tend to, regardless of age, occupy more of this globally disconnected state. Okay. And just a few more. We can also use these approaches to differentiate between subtypes. Here we're looking at Alzheimer's disease and subcortical ischemic vascular disease. And here what we find is that the transition time between state 2, again a weakly modularized state, and state 3 is significantly different between those 2 groups. They have more transitions in the AD patients than in the SIVD patients. And then the last example is just sort of a broad case of substance use data. We divided this data into multiple groups, controls, individuals that are only alcohol users, smokers, combining with alcohol and smoking, etc. And that we find some really interesting patterns occurring. So for example, if we look at these states, state 5, for example, is kind of broadly different and barely almost in the marijuana group across all the different sort of substance use categories. State 1 is more specific to smoking, for example. This is an attempt to try to summarize a little bit of what I showed you. Again, looking across a bunch of different disorders, trying to see if there's some specificity to these changes that we're seeing. And it looks like there might be. This is hardly comprehensive, and I think we need a lot more work to try to synthesize. There's a lot of work out there these days already in the dynamic connectivity world. Now, a few examples of more advanced metrics, and I'm just going to scratch the surface here, but there's a ton of interesting approaches out there. You can, for example, look at time frequency analysis. Here we're looking at an approach based on a Morlet, complex Morlet wavelet transform. Essentially, we have 3 different things we can look at. We can look at the amplitude of the dependency, and so that's the intensity of the color. We can look at the spectra, high frequency, low frequency, and we can look at the phase. So red is correlated, blue is anti-correlated. So there's kind of a lag difference between them. And this is interesting because we find, for example, some cases where two states have a very similar amplitude profile, but a very different spectra. And one of them differentiates based on, say, a cognitive score and the other does not. And it's not separable unless you can incorporate this additional information. There's also been a lot of talk about different approaches. Should we use, what window size should we use, different windowless approaches, instantaneous approaches, etc. Here's one example of a windowless approach in which we're using a dictionary learning algorithm. And so the idea is that rather than capturing a state that maybe is the blurred version between, there's only two states here, but it looks like three, for example, we want to allow for sharper transitions to be captured while mitigating against noise. And so here you can see, compared to the ground truth, the approach that we used is a pretty well captures it, where a sliding window does more poorly on in particular, the ones that have rapid transitions. This is just a slide kind of mentioning that dictionary learning approach. We're capturing these different states here, assuming that there are a limited number of states per individual. So there's a sparsity assumption there as well. Another approach based on sort of a signal processing worldview is to basically say, well, a window is essentially a filter on your data. And we can actually mathematically map the relationship between the activity time course and the connectivity time course given a particular window profile. That's a filter. We can then create a bank of filters to capture all the possible windows at different levels, different frequency bands, for example, and then estimate that entire system at once on the data, so we're not specific to one window. And so then the output, in this case, I'm showing 8 states estimated from the data, and then we have the weights associated with the different filter banks. And so you can see in this case, we have a very low frequency one. Here we have one that tends to be more high frequency. And we can also quantify, for example, group differences in this type of an approach. We can look at trajectories. So here we're looking at energy, which is essentially like a density-based approach. We're trying to find dense portions of time, where there's clusters essentially, at which point then we can create movies around that point in time. And so this gets at more of the trajectory. How is this behaving in this sort of, in this case, two-dimensional space? Here is an example where instead of using just the correlations themselves, we're using the correlation and its first temporal derivative. So we're trying to get at information about flow into and out of that state at any point in time. And so we have a pair of matrices now, the main correlation and its temporal derivative state. And we find when we do this that we get sometimes these clusters, right? So we get groups of state A, which contains 2 different clusters of the paired states there, which represent sort of antagonistic relationships on the derivative side here. So we have similar amplitude profiles, opposite or negative derivative profiles. So we can model this as sort of an attractor type model where we have two different attractors and the flow is moving into or out of or away from these different attractors. And so using this type of an approach, we find some really interesting results if we look at traumatic brain injury in a longitudinal study. This is data from Hans van der Hoorn. And so starting from one state here, if we look immediately after the injury, or 2 weeks after the injury rather, we see that the attractors are disrupted. 4 weeks after the injury, we see some attractors recovered, and by 3 to 4 months, we see full recovery in the attractors. And so that was quite striking to us and seems like a really interesting way to model these data. This is sort of a catch-all slide to kind of say there's a lot going on in this dynamic data. There are lots of ways to analyze it, as you've heard from these other talks in the session. But another way we can do this is we can kind of search through that dynamic space for things that are repeating, right, consistently. And so here's an example using what's called tri-clustering. So we're basically clustering on the connectivity values, the time course, the transients over time, and the subjects. So we want to get more homogeneous subsets of the data, at which point then we can start to do comparisons within those subsets. And if we do this, we see it's much more sensitive to differences between patients and controls, for example, because we've got a lot less variability when we do these, when we look at these subsets. We're kind of comparing apples to apples, so to speak. Here's another approach based on deep temporal convolutional neural networks from Hiroshi Morioka and Apo Hyvernen, and looking at nonlinear ICA to capture temporal primitives from the data, which are occurring at different points in space and time. And so you can see 3 different examples of those, as well as their profiles in frequency space. The last 2 examples I wanted to kind of indicate are sort of multimodal type examples, or different kinds of data beyond imaging, beyond functional resting fMRI. Here's an example where we incorporated single nucleotide polymorphism array data. So we have a bunch of SNPs. And what we did was use something called parallel ICA, which I'm not really going to go into detail on, but essentially we're looking for maximally independent components that are correlated to one another through the states as well as the genomic data. And we find a pair, a coupled pair, when we do this, that was linked to a SNP profile. So here we have state 1, and that's the same state I showed you earlier when I gave you that first schizophrenia example, where healthy controls occupied state 1 significantly more than patients. And then this is the genomic profile here. And if we look at this in the context, if we can compute what's called the polygenic risk score from the component, the genomic component that came out, and then look at its correlation with state 1's amplitude across subjects, we see a strong and strikingly negative relationship. So the risk for schizophrenia is lower the more time that these individuals are spending in state 1, which was where the healthy controls were spending their time. So this is, again, it's consistent convergent evidence that suggests that we're learning something meaningful here. And the last example, excuse me, is linking brain structure to brain function. Again, using a deep learning approach. Here we have an, I'll just sort of break it into 2 parts, an attention model, where we've got figures and captions, and you're trying to mine those. And essentially the idea is to highlight for any given word where in the figure is relevant to that word. For the alignment model, these are used for translation, so where we might have one word mapping to 2 or more words, or the order might be different. And when we do this for brain imaging, we're thinking of the structural data as the image and the dynamic state vector here as the caption of the figure. And for the structural features, we're using ICA to get at covarying structural networks. So the model is here, where we have these structural networks kind of coming in at the bottom. At the top, we have the dynamic state vectors, and we're trying to estimate what's called alignment between the two of those. And we do indeed find strong alignment, perhaps not surprisingly, between brain structure and brain function. Here you can see it's well above 0, highly significant in both patients and controls. But we also see, interestingly, healthy controls are significantly more aligned than schizophrenia patients in this middle temporal gyrus structural component. And this is state 3, showing a relationship here also with cognitive scores. Okay, so in the last couple slides, I just wanted to mention software. So we do try to release all of our software and tools. Most of the approaches I mentioned have been already released in this GIFT software. The ones that aren't there will be there soon. And so this is a MATLAB toolbox, but we also release an executable version so you can compile to various operating systems. There's a Python interface called GiftPy, and there's also a containerized version of Gift. If you want to just learn a little more about dynamic connectivity, we have kind of a broader review paper here, which is online. This isn't focused only on the dynamic FNC approaches I mentioned today, but also other approaches, for example, CAP scores, etc. And it also introduces this dynamic FNC toolbox and GIFT, which has a lot of different techniques and tools, ways to visualize the data, explore the dynamics in your imaging data. With that, I will close. This is a picture of some of the folks at the Trend Center in Atlanta, without which this work wouldn't have occurred. And also thank you to the funders of the work as well. Thank you very much.